what happens in Huntington's disease is through replication errors, these two trinucleotide repeats increase in number, and that ultimately, for some unknown reason, leads to a big neural degeneration. There's no cure for it. You can do a genetic probe test to confirm whether you inherited the mutation if it runs in your family, but there's no cure. So would you want to know? Would you want to know you're going to die in your mid-40s or 30s? I don't know. But that test does exist. So real quick, we're going to screen through these pedigrees here. So we all recognize in the lab the pedigree shows a family history, females and males, and their offspring, and then their in-laws, and so on. Typically, the trait is shaded. This is phenylketonuria. This is a inborn error of metabolism. We see that in this case, we see a female and a male. With two of the grandkids have this condition, and based on the pattern of inheritance, you can often determine whether it's autosomal dominant recessive or X-linked, like we did in lecture. This is a autosomal recessive condition, because if this were dominant, there's no way these parents could produce these kids, for example. Achondroplasia, this is leads to dwarfism in humans. Here we see this is an autosomal dominant condition, where if one of your parents has it, at least some of the offspring are going to have it. Okay, not guaranteed. If in this case, father is heterozygous here. Okay. Duchenne muscular dystrophy. This is one of the X-linked recessive traits. The clue is males exclusively. Okay, and this is one of the very rare X-linked dominant traits. These are very, very rare. This is the only one I know about. This is a disease that leads to defects in tooth enamel, and it's hard to detect through pedigree analysis, but typically you have males and females in roughly equal proportions in the pedigree analysis. One of the more famous studies of X-linked recessive traits come from the royal families of Europe. This is Queen Victoria here. She was one of the longest-lasting monarchs on the English throne in the 19th century. She ruled England when they used to say the sun never set in the English Empire. Remember when I was in Bangalore, India, they had a statue of her downtown Bangalore, because that's back when India was in the realm. And this is a family portrait in 1894. A lot of, of course, if you're at that level of society, especially in the 19th century, you marry, you're encouraged to marry others at your level for political purposes. And maybe this is the circular. I'll point out two here that I recognize. This is two with the feathered boas there. There's Victoria. This is her granddaughter Irene, who married into the Prussian royal family, and this is her granddaughter Anastasia. It wasn't Anastasia. Oh, what the hell is her name? Princess Alexandra. Sorry. Next to her husbands are Nicholas II of the Russian royal family. So she was the Tsarina. What's interesting about it is that Queen Victoria was a carrier of hemophilia. And both of her these two granddaughters were also carriers. In fact, Victoria introduced, the English family through intermarriages introduced hemophilia to most of the royal families of Europe. Here's the pedigree here. So on this particular, they're using a green circle with a dot to indicate a carrier. Now carriers are healthy, right? That means she has one X chromosome with the mutated hemophilia gene, the other one is a normal gene, so she was lived a very long life. Her husband didn't live that long. So if you go to England, almost every other structure is named after Albert. Royal Albert Hall, Albert Park, blah, blah, blah. They did well in the kid department. They produced a lot of children here, a son that didn't survive birth. Let's see, 1, 2, 3, 4, another daughter didn't survive. 1, 2, 3, 4, 5, 6, 7 kids. 2 of the daughters were carriers. One son was a hemophiliac. And then here's Irene, Princess Irene, married to the Prussian royal family. 2 of her sons are hemophiliacs. There's Alexandra, the Tsarina. Of course, she had 4 daughters, and their crown prince, Alexei, suffered hemophilia. Of course, these were all murdered by the Bolsheviks in 1917, the Russian Revolution. But you can see how hemophilia from one carrier, sort of matriarch, spread throughout the royal families of Europe. Okay, Notice a lot of these males didn't produce offspring because it's hard to survive hemophilia, especially if you're a boy. How long does it take a boy to fall down and scratch his knee? Oh, about 5 seconds. Now, the different severity of hemophilia, you're not going to bleed to death right away, but it does compromise your health. Some did survive. Of course, these were very special boys, so they were nursed as best they could. All right. Now, chromosome structure can vary because of sort of eras and maintenance of the chromosomes. We talk about mutations like a base misspelling, what I refer to as a SNP. That's often through a replication mistake or a spontaneous mutation. You know, at your high body temperature, mutations happen spontaneously. And I'll talk about some of the more important mutations that occur commonly in the next chapter. Fortunately, and the mutation rate's rather high, but fortunately, most of those mutations are corrected by your DNA repair mechanisms. Your cell spends a lot of effort to monitor your DNA and correct those mutations. So there are misspellings in the DNA, but these are structural alterations where it leads to a loss of DNA or a gain or an extra copy or a change in the structure. Either a piece is broken and attached in the wrong orientation, what's called an inversion, or sometimes pieces of chromosomes are traded between other chromosomes, what's called a translocation. A deletion is when a part of a chromosome is lost. Okay? And that can result from a common genetic mistake called a depurination, where a base is lost, and then the repair mechanism just kind of stitches it back together and loses the piece. And you can imagine if this is a critical gene, a deletion would be a very serious issue. However, in the human genome, most deletions, not a big deal, because of your roughly 3.2 billion base pairs that make up your 23 chromosomes, okay, how much of this DNA actually codes for genes? Well, about 1.5%. When the human genome was planned at the end of the 1980s at Asilomar in Monterey, when all the great molecular biologists got together and decided we want to sequence the human genome, they took bets on how many genes were in the human genome, how much the DNA was critical for functioning of an organism. A lot of the bets were, 100,000 genes, 50% of the DNA's coding carries information to build proteins. They were all way off. The number of genes is somewhere around 21,000, and 98.5% of the DNA doesn't code for anything. It was known as junk DNA. It's not really junk, but a lot of it is non-coding. So if you have a mutation, a base pair change, or you lose something, it mostly happens in this junk DNA. You can survive. So that a lot of that junk kind of buffers what could otherwise be deleterious changes. Okay? That's in contrast to bacteria. Prokaryotes have much smaller genomes, and almost every part of the DNA of a prokaryote is coding. They don't have any junk. Okay? They're producing a new generation every 30 minutes. They can't afford to copy a lot of extra DNA to pass on to the future. But the downside is if your entire genetic molecules are critical and you have a mutation, well, you're screwed. Okay? With all this non-coding DNA, deletions, misspellings, you can buffer a lot of that. But rarely it happens in a critical gene and you're screwed. So deletions often due to spontaneous mutations that happen just because of your body temperature. Okay? Random molecular, at 98.5, a lot of molecules moving pretty fast, and sometimes, chemicals change shape, they break apart. So spontaneous mutations are part of the game. Plus, we live in a very mutagenic world. Cosmic rays from space, dangerous chemicals, both natural and synthetic. And so these can all lead sometimes to a deletion. But again, a lot of the junk DNA, it's not really junk DNA. It has much of it has function. A lot of it actually serves as software to control gene expression. Okay, and that code is being unraveled now. So even though the human genome has been deciphered, a lot of it it's unknown what it does. Okay? It's unknown what a lot of that information says in terms of making a human work. So deletion is a loss of a gene. Structurally, you lose a piece of DNA. Now, on the other hand, sometimes a new sequence of nucleotides is inserted into a chromosome. Okay? Either through replication slippage, or it's been transferred either Or sometimes in a virus, you get infected by a virus. Viruses, when they infect certain viruses, when they infect a cell, they insert their viral DNA into a cell. It becomes part of you. There are a lot of viral genes. When people study the human genome, they found hundreds of examples of viral insertions of our past. Okay? That represent past infections. And so you can have an insertion, it's not on the slide there, where a new piece of DNA is added to a chromosome. And collectively, insertions, deletions are called indels. You study, if we get into genomics, you'll see this term indels. This is when people compare genomes amongst individuals, they see that, well, you've got an extra piece in your DNA, in this chromosome 8 that I don't have, and over here, chromosome 2, I've got a piece missing that you have. So insertions, deletions, the result of these structural changes. And they're part of the structural changes that make humans different from each other. A duplication, check this out. Now, in some cases, we see, again, these are just letters to give you an idea of the sequence. Here we see D and F has been repeated. Okay? Now, often this happens from replication slippage, where during the replication process, the machinery slips and then recopies the piece again. It also could happen from mistakes in recombination, where sometimes instead of a reciprocal change between the maternal-paternal homologs at prophase 1 in the tetrad, sometimes there's an unreciprocal sort of recombination where one homolog gets more than the other. Now you've got like extra sets of a particular gene. So a lot of these things are happening through normal processes. There are sort of mistakes that happen, but they can lead to duplication. Now any change can be serious if it compromises a particular critical sequence, but duplications actually contribute to long-term survival of a species because they provide new genetic information that can lead to new traits in an individual. Like if I've got 2 copies of a gene, one can maintain the critical, if these are